You'll notice there are no slides. George has volunteered to do shadow puppets in front of one of the projectors during my talk. The organizers actually asked me to combine two activities with this talk, and one was to summarize the workshop that was held at NIH for the last 2 days on diet and microbiome. Just for a show of hands, how many people participated in that? Wow. Fair number of you. Okay, so you two folks can take a nap, and I'll tell the other people about this for a few minutes. The background on this was that it's quite clear that the microbiome folks who are experts in that area don't seem to know a lot about nutrition. And the nutrition experts don't seem to know very much about the microbiome. And the idea really was, since diet is the primary food for our microbiome, how can we, not necessarily standardize, but how can we account for that in vitro, animal model, and human clinical studies? And there's minimal information given in most studies on this. You'll see in a mouse study, cellulose was the fiber in our diet. Well, there are 12 commercial types of cellulose available that vary markedly in particle size and water holding capacity and so on. And obviously, there's a difference in fermentation. Even though cellulose isn't well fermented, microcrystalline cellulose isn't fermented at all. Various types of powdered cellulose can be fermented. I've seen numbers of 10%. It might be higher under mice that have different microbiomes. And that's one of the problems when we buy our mice from Harland or from Charles River. Those mice aren't all the same, even if you're buying Swiss Webster mice or C57 blacks. You know, they're raised differently. There's genetic drift among these animals, which caught Charles River by surprise about 30 years ago. But each supplier of animals has their own diet that they stick to. And Justin Sonnenberg has shown that over generations, mice will lose certain classes of bacteria. So you're not necessarily starting with the same organism, even if your paper reports that. And it's the same thing with the diet. Devin Rose asked folks at the workshop, how many people who've done human studies have reported, we've used normal healthy volunteers who haven't been exposed to antibiotics for the last several months on an uncontrolled diet, and pretty much every hand went up. And that's part of the problem, because we've all got our own fingerprint of a microbiome. That doesn't necessarily mean it's so complicated that we can't study it, but we have to take these variables into account, and it's the same thing with the diet. George gave a wonderful opening keynote talk to that workshop, and I almost held my head in my hand saying, oh, this is even more complicated than I thought it was, because we have chemical differences, structural differences in the various dietary fiber sources. And again, it doesn't mean we're paralyzed, that we can't move forward, but it does mean that we have to take into account these variables and either increase the number of subjects or increase the amount of analyses we do and increase the reporting we do for these studies. And so for the federal agencies that co-sponsored this, the idea really behind this was to improve reproducibility and rigor in these studies. In Washington, we're all news junkies, and so over the last couple of years, there's been repeated reports in the popular press and in the scientific media about the lack of reproducibility in scientific studies, whether it's human trials or animal models. And that just translates to really bad publicity among the tax-paying public, who pay, if you've got NIH or USDA grants, they're paying for your studies. And so people actually have, the public actually should have a say in this. And we're very sensitive to the optics of the lack of reproducibility. in this. And part of that lack of reproducibility is the lack of detail. You know, when I was a graduate student 45 years ago, I was taught that you have to have enough in your methods section that somebody reading your paper can reproduce your experiment. There isn't a paper published today that I think provides that level of detail. So we had a number of sessions that were divided into basically the components of diet that affect the microbiome, and it's primarily fiber and resistant starch. We had some really excellent talks on that to introduce the subject, and actually all the speakers are here, so I don't need to belabor that. We had a talk, or a series of talks on animal models, people mostly using mouse studies. Kelly gave a really good summary of the non-rodent models that are available, and you know, there's a lot of debate about the mouse has a big saccular cecum like all other rodents. We don't. You know, do they inherently need lots more fiber than we do? And is a humanized mouse really a valid and better model for humans? And we don't have any answers to any of this. Andre Marriott from Laval talked about the effect of polyphenols, and there are hundreds, if not thousands, of polyphenols that we consume, most of which have antimicrobial activities, most of which the majority actually makes it to the colon. Benoit Chassin from Georgia State talked about emulsifiers. This seemed to be fairly controversial. There were very little comments about that. I don't think most people accepted it, but I can tell you that in the early 1970s, the late George Mann wanted to study the effect of emulsifiers on serum lipids, and he ended up studying the Maasai in Africa because he couldn't find an American population that wasn't regularly exposed to emulsifiers in 1970. We sort of built on those animal studies to actually go to in vitro models, had a couple of talks on that, being more and more studied because it eliminates the host variable, which Karen just talked about quite a bit, and whether or not you can study defined populations, like a community of 10 bacterial strains, rather than the hundreds, if not thousands, that each of us harbors. And all of those studies led up to the final session yesterday afternoon on human studies. And they're even more complicated animals to work with than mice or rats or pigs. Not sure we have any solutions. We will publish eventually a summary of the meeting that will hopefully get at some best practices for design and reporting of diet and microbiome studies. So in the remaining minutes I've been tasked with, as George said, talking about the future. And for those of you who are not from the United States, Yogi Berra was a very famous baseball player. He was Hall of Fame catcher for the New York Yankees, and I grew up in New York City, so he's my guy. And Yogi was a very quotable person, and he said, making predictions is really hard, especially when it comes to the future. So don't hold me to this. But I think the diet and microbiome is one of the real future areas for the fiber business. You know, if I had to invest, I would be putting a significant part of my retirement into microbiome studies. There were a number of points made by the earlier speakers, and I'm not going to tell you you should have listened to Joanne because you should have, but she made a number of points. Stool weight is an important biomarker, so you get a brown star for that, Joanne. The Bristol stool scale, you know, it's really simplistic, but has a lot of valuable information. In fact, there was a paper in Nature a couple of years ago looking at a few thousand Belgians and Dutch people that the Bristol stool scale was one of the best correlates with dietary intake. And you can buy a coffee cup with the Bristol stool scale on the side. And I have to say, coffee has a very significant impact in some people on the consistency of your stool. Joanne also mentioned that short-chain fatty acids yield about 10% of the energy in most humans. And one of the questions I have about that I don't think there's any answer for, but I have concerns about, is whether or not those short-chain fatty acids actually are utilized with the same efficiency as metabolism of macronutrients further up in the small bowel. And all the physiologists tell me that I'm just crazy to think about this because the GI physiologists are always right. And they tell me that all the butyrate is used by the colonocytes, which we know isn't true because there are G protein receptors in tissues everywhere, including the central nervous system, that why would there be a receptor for butyrate in your brain if some butyrate wasn't getting there sometimes? Not saying that there's short-chain fatty acids coursing through your bloodstream right now, but after lunch, there might be a little bit. So Kelly talked about fermentation of roughage. I was really glad to see that term there. Some of you know that I worked with Dave Klurfeld for 15 years, and he was one of the original organizers of what was called the Washington Spring Fiber Meetings before it was named after George Vahouny. And Dave actually did the very first paper showing the biological effect of dietary fiber before it was called fiber. And he showed that dietary fiber added to the diets of rabbits lowered serum cholesterol. And his lab group extracted hundreds of pounds of rabbit chow to get to that non-digestible portion. And this is one of the problems that dietary fiber research is a relatively young field. As a matter of fact, somebody, I think it might have been Kelly, pointed out that or maybe George, that Burkitt Trowell Painter really put this on the map. And that was only in the mid-1970s when it was beginning to be accepted. And that was Dave Klurfeld's paper was in 1964. He told me it was the hardest paper he ever had accepted. It took him almost 2 years to get it published because everybody knew fiber was inert, had no biological effect. And in fact, that's one of the problems the microbiome has. For decades, everybody knew this black box in the colon didn't do anything. It was past the point where there was any absorption. We were only absorbing water and electrolytes in the large bowel. And in fact, when Dick Calvert was a postdoc shared between my lab and George Vahouny's lab in the 1980s, he showed that wheat bran enhanced bile acid absorption in the colon of rats. So we've known for decades that this happens. It's just been generally ignored. Kelly mentioned Jeff Gordon's studies on obesity in mice, and they're beautiful studies, and they really catalyzed the thinking about nutrition and the microbiome. But it was a really simplistic hypothesis that 5 times has been reproduced and 5 times has been refuted. So I still don't know the answer whether or not the phyla of Bacteroidetes and the ratio to firmicutes really matters in this at all. And it might be just too attractive to be true, but he might be right. Jeff's a lot smarter than I am. You know, the pharmaceutical industry has embraced genomic microbiome differences and individuality. We, of course, in the food industry don't have the profit inducement that the pharma industry has, but there are likely analogous effects for food that can be teased out. So I think Karen made the point that we still have mostly associations and not causality, and that's what we can achieve with animal models, I think more so than in humans. But clearly, we need to go to humans to maintain credibility. The inflammation that Karen talked about is an area that I think can also contribute to thinning of the mucus layer. A lot of people study panels of inflammatory markers, usually in the serum or in fecal water or something. And in one of my part-time jobs, I'm also associate editor of the American Journal of Clinical Nutrition. So I see this literally on a weekly basis. Somebody will feed a diet to volunteers and do a panel of 25 inflammatory markers. 2 will change significantly, and they'll say, this proves that treatment X is anti-inflammatory. And my response is, You know, it didn't work for 23 or 25 markers, but we're in love with P less than 0.5. So I really liked one of Karen's closing ideas of matching fibers, plural, with the host microbiome. I think that's where the future is. You know, in the last couple of days in the workshop, we had a few people talk about how the food industry is making these low-fiber foods that don't feed our microbiome. Well, in fact, I think this is the opportunity for the food industry to provide foods that people will buy more than once, that are high in a variety of fibers that can feed our microbiome. And of course, the question is to satisfy Paula and her office that you're having a health benefit, not just that you're causing a bloom of certain species of the microbiome. And the key is linking microbiome changes to a health benefit. And that isn't cheap, and it isn't a short study. So I've intentionally left a few minutes for debate about this, and I welcome anybody to come up to the microphones and disagree with me.
Wouldn't you know it'd be Stuart?
Yeah. Surprise, surprise.
Yes, it was an impressive session, and I'm Jan-Willem van der Kamp. I'm from Netherlands, you know, and I'm involved in a big European project about the microbiome. My New Gut is called, and one of the aims of the project to make links between public health dietary recommendations and the microbiome. And if you look at the past and you see dietary recommendations and you type in microbes and things like that, the only thing you find is infection. Because that is the only thing where in current dietary recommendations the microbiome is related. And in some, there was one exception, and that was the USDA scientific report for the new recommendations. And there was mentioned that a study was done of microbiome could have some impact on dietary recommendations, and the answer was not yet. Maybe in the future, and maybe you are at USDA ARS. The one direct question is, will this be the case in 2020? I think, I think that is indeed, because you have indeed what you said, you have the microbiome community, they are enthusiastic, and those regulators who make the direct recommendations say, okay, it's good for the microbiome, so what? But what is, I was impressed indeed what Karen was saying, but the thing, a lot of correlations, but where I think now comes the conclusion became a question mark about the hard relationships. But looking in the future, what is your view? Is 2020 already the time that this correlation can be made, or do we have to wait longer?
Early in my career, I thought things would be solved more quickly. You know, when I was a postdoc, I thought just increasing beer and beans in the diet would solve all of our problems. And I've been trying to do that ever since for personal change, but hasn't worked out quite that way. Really, I think the only therapy we have that is semi-validated is fecal microbial transplant for C. difficile diarrhea. That's really the only payoff so far. I wish that we would have something for 2020, but I would really be surprised. You know, I think the Human Microbiome Project, which was a $175 million investment by NIH for the last 10 years, has really focused in this first decade on who's living in the gut. And I think it's far more important to know what they are doing in the gut. And that's metagenome analysis, that's a lot more expensive. You know, I think just like genomic studies, metagenome prices will come down right now. You can't afford to do adequate numbers of those studies on most research grants. So I'm thinking, and then we have the issue of the talent to understand the bottleneck of data reduction. I think that's one of the most difficult areas right now for microbiome analysis, is getting the right programs, and we'll develop that, I think, through machine learning to understand how the correlations predict changes in health. And I'm hoping it'll happen in 10 years, if you want a number. Stuart.
David, thank you, as always. Actually, so your comments made me think about an article I read recently about bias, and how, as scientists, we all have our bias, and we can take the same set of data and draw quite diametrically opposite conclusions depending on the bias coming from. So thinking about your comment on dietary fiber in its early days when it was thought of as inert, it was obvious to everybody that it was inert, and it took the tremendous effort, personal effort, to change that perspective. What do you see today as biases that maybe we all have, or that many have, that looking to the future you think needs to change?
Wow. There's a long list. But I'll tell you, one of the biases that I see, I'm on NIDDK's advisory council, and they're the largest funding unit at NIH, largest funding unit of nutrition research at NIH. They're one of the smaller institutes by budget. And I see this every 3 months when I go for the advisory council meeting. There's a significant bias, even among their advisors, against nutrition. It just is not sophisticated. We're in the grind and find mode still. You know, there are researchers on pancreatic biology looking at the molecular mechanisms, the genetic control of beta cell biology. We're not doing that in nutrition studies. You know, we're still not quite in the home economics era, but we're viewed by the outside world as being home economists in a lot of ways. And Barbara, being a former nutrition chair, is making faces, but I was also a former nutrition chair, and I saw that at a lot of meetings. Not everybody is in that mode, but I think that is the outside view of the nutrition field, that it just is not very sophisticated. We're feeding this relatively uncharacterized stuff to people. And in fact, the reason for that is the USDA's food composition database, which my program has, is just not that good for types of fiber. For instance, we've got total fiber for a lot of foods. We have soluble and insoluble for a tiny number of foods. That's it. If you listen to George a couple of days ago, we don't have anything about the structure of fiber. And maybe we need that. You know, so there's, and then you heard yesterday about the bias of assessing what people eat. It's more of an art than a science.
Sounds like a long list. Thank you.
Dan.
Dan Geller, University of Minnesota again. Thanks for bringing up the issue of lack of reproducibility. I think that is a huge issue. I think part of it is diet design. We did an experiment some time ago replicating someone else's work. A woman named Natalie Delzine, who's done some great work feeding high immunolome levels and seeing this dramatic increase in PYY in the plasma. We replicated that, but when we added just a little bit of another fermentable fiber, just a small amount, completely eliminated the effect. So I guess it's two things. Replication is important, and two is using more realistic diets. Feeding single fibers is great. I've done it myself a lot, but it causes a lot of problems. And I think we have to remember that We don't eat single fibers.
That's right.
Mixtures of fibers.
That's right.
We should be considering that when we design the diets.
Great point.
As always, I have to agree with everything Dan said, but I want to point out just another personal experience like Dan had about reproducibility. About 30 years ago, I had a postdoc in the lab, and we wanted to reproduce a study that had been published in the Journal of Nutrition, and we used identical diet. We used a different strain of white rat and got not just a lack of reproducibility, but exactly the opposite of what was published in the Journal of Nutrition. And I almost kicked the postdoc out of my office. And I said, Eugene, you got to go back and do this again. And he came up with exactly the same findings again. And we could never explain why we had not just a lack of reproducibility, but a 180-degree difference in the results. Barbara.
Great. I think what I have to say builds on what Dan and you just said as well. If you think about where we are with dietary fiber, it in some ways reflects where we are with dietary recommendations. You know, is it the single substance? Is it the dietary pattern? And I mean, I can even look at what Eric presented earlier, the two diets he compared and said, no, you're looking at a dietary pattern. You're not looking at fiber. And if you think about FDA's dilemma in how is it going to define fiber for nutrition labeling, does it have to be intact, intrinsic? Can you find a way to use isolated synthetic? So this fact that we're in this tension point between is it a dietary pattern that is the way we want to go forward and make recommendations, and then how do we do that versus each individual component. So when I think of the future, I think about how are we going to get better at trying to understand that dietary pattern and what are the key essential components of that. The other area that I tend to think about, one of the things that struck me at the microbiome workshop and in the presentations today is the power of the methodologies that we now have. And you know, you and I attended these before they were the Vahouny conference, so sometimes it can look like, gee, we're looking at the same questions. But when you look at the power of the methodology that we now can bring to bear, or that scientists can bring to bear, my hope is that it gets us to a new place. Either we need to change the paradigm we're working with, or we are able to resolve some of those issues because the drive to the methodology that helps us answer those questions has been there. So the future's always brighter, right?
Thank you for those comments, Barbara. One point I want to make is that we're sort of chained to our pasts. Almost all of us in the room were probably trained in the reductionist school of science, where we try to isolate the one component. You know, everybody accepts that all these chronic diseases that are related to, putatively, to low fiber intake, to high other nasty foods, is multifactorial, multi-stage. But we're looking for the one change we can make to reverse all of that. And so I encourage you all, this paper that's probably more than 10 years old now, written by Tom Robinson, who's a pediatrician at Stanford Medical School, on solution-oriented approaches to obesity. And he talks about, don't focus on one thing. Focus on, and then this is probably why, one of the reasons we haven't solved obesity, is that we're doing the reductionist approach. You know, we need to know if it's calories or exercise, but we don't want to look at them together. You know, we've got to separate that effect. And each of these dietary effects probably isn't strong enough to have that impact. So we've got to change multiple things at the same time, and then you'll be criticized for not knowing what helped. So I think that's the end of our time.